Good morning, or good afternoon, or good evening, everyone from the world. My name is Zhugen Yang. I am a senior lecturer in sensor technology at Cranfield University in the UK. Today, I will talk about CRISPR-Cas-enabled paper microfluidic device for rapid diagnostics. For people who don't know Cranfield University, which is an exclusively postgraduate student university based in the north of London, which is around 1 hour away from London, Cambridge, and Oxford. So it's quite easy to access. So as you know, there's a lot of challenges for pathogen detection, either for biomedical diagnostics or environmental and public health or food safety. This is a very important detection techniques for those pathogens. Conventional detection method relies on culture method, which is time-consuming, takes days or weeks, and less sensitive. There is a very, very popular method, which is molecular method, especially over the past several years of pandemic. Everyone now knows PCR method for testing of SARS-CoV-2 viral genetic material. However, this technology is very much relies on the central laboratories and well-trained skilled persons to carry out the sample processing, analyze, and data interpretations. So it's not always useful or popular in the resource limiting areas. So there's to address these challenges on the pathogen detections, there is a great need for rapid, low-cost, sensitive, and a portable sensing platform enabled to rapid testing these pathogens or for all those applications. So I will give you some our recent work on our development on the paper-based device or paper microfluidic device for rapid diagnosis, coupled with molecular methods such as isotope amplification and CRISPR-Cas-enabled molecular assay to improve the sensitivity and the specificity. Finally, I will give you some more recent work on the wastewater surveillance as a new public health tool to address infectious disease challenges. When we talk about paper microfluidic device, we always want to mention science hero, Professor George Whiteside from Harvard University, who has pioneered a paper microfluidic device more than a decade ago. So he firstly proposed paper as a substrate to fabricate electrodes and make a new glucose meter for monitoring diabetes. There's a lot of applications emerging in both as paper microfluidic electrodes or paper as a sample preparation steps for the different sensing application. In our approach, we develop a uniquely folding and unfolding paper device, which is wax printed to formulate hydrophilic and hydrophobic layer, which can be used as sample preparation steps. So as you can see from here, it's mainly composed of 3 parts. One is a paper-based device. So we have the black layer, which is wax, can be hydrophobic. And this white layer area is hydrophilic. So that's we can use as a fluidic channel to process these samples. And we also have a glass fiber, which can physically absorb and adsorb nucleic acid because of interaction of electrostatic interaction. And we also have a plastic plate, which can accommodate the isotope amplification in order to improve the signals. So what we do is in our platform, we will first introduce samples into glass fiber after lysis of these clinical samples, which might be from blood, saliva, or semen samples. Then this glass fiber can electrostatically absorb this nucleic acid. Then we can go through the washing, using washing buffer to purify those DNA material or RNA material, but wash off the non-DNA residues. So afterwards, we can elute those genetic material from glass fiber and go through this 5-spots or 5-star microfluidic channel into this plastic plate. Then we introduce the reagents to make the isothermal amplification happens. So basically, we can use either loop-mediated isothermal amplification or coupled with CRISPR-Cas-enabled amplification. So the whole assay takes less than 1 hour. That's pretty much fast and enabled sample-to-answer analysis. So we've initially developed this method for the veterinary diagnosis. And maybe you still have some confusion on this workflow. I have a very short video to let you go through this process. Okay, this is images. What we captured within the field testing of paper-based device in a local Indian farm. So basically, we tested a small cohort of semen sample for the veterinary infection disease diagnosis. So we can either read the results using naked eyes for the calorimetric change, or capture the image to do the semi-quantification with those 3 pathogens relates to this sexually transmitted infection disease of bovine. So this field testing just give us confidence to use as a testing case in the field. We further implement this technology by changing the design of this paper microfluidic platform, and also improve the detection method from the calorimetric detection to lateral flow device. Sometimes there is background light can have tiny influence on your results. So we firstly develop this method and test a small cohort of sample in our lab. And together with collaborators, we travel to local primary school in Uganda in Africa. We test malaria samples from local school students, and we compare those results with the gold standard RT-qPCR method. As you can see, we have a very competitive sensitivity and specificity. This is another example. We show this paper-based platform coupled with RT-LAMP, which is because we are targeting hepatitis C virus, HCV. We're using a RT process to do the isotome amplification. And also we're using this lateral flow device to test the positive and negative results. We can also test the viral load by this optimization of the detection results. We developed this method and also validated this in the lab-based samples. Then we go for a cohort of clinical samples showing very promising testing results, which is validated by RT-qPCR method. Here is another example. We in collaboration within a group from University of Edinburgh, we published results and further improve the molecular assay together with the Cas-9 platform. We call it is AGATHA viral RNA sensors. So we basically design a new mechanism independent within the viral RNA. We can also trigger this cleavage translation, cleavage, and then using this Cas9 as a Cas enzyme to improve the sensor. We prove this concept and using a mobile camera to capture the image for calorimetric detection. So this is just to show us we can flexibly detect the RNA sensor for different viral pathogen detection. To give a brief sum up, based on this origami paper sensor, it has a lot of advantages, such as it's very cheap, which less than 1 pound, including reagents and a device, as well as rapid, which can provide a sample to answer detection within an hour. The device itself is very user-friendly. As you can see here, the picture which I took during the trip, my Indian trip, after training the local student within within several hours, they can run this device independently. So this is extremely useful to test the pathogens in a field for point of use or point of need analysis. So we have proved and demonstrated a few applications using origami paper device for pathogen detections. However, over the pandemic, there's a lot of new technology for this rapid diagnosis of infection disease, especially coupled with CRISPR-Cas technology. Here, I want to introduce you some new concepts as a public health tools. Last year, Science magazine has summarized 125 ground questions for the exploration and the discoveries. One of them from the medicine and health sections: can we predict the next pandemic? As the international travel, climate change, this potential pandemic may come up again, or especially for some new variant. If we can make us much ready for the future pandemic, that will save us a lot of economic loss and money and people's life. So potentially wastewater surveillance could be as a potential tool for this public health management. Firstly, let's explain you what is a wastewater surveillance. So the concept is that each people will shed viral particle or pathogens into the toilet if we get a virus or infections. So this virus will travel into the toilet, end up in the wastewater treatment plant. If we collect sample to analyze those viral particles, so this will provide extremely early information that even before the symptom developed. Here is a time window show you even people have mild or no symptom, they still shed this viral particle into the wastewater. Basically, we can monitor them and give us a early warning information for this transmission of infection disease. Typically, it can be 5 to 7 days earlier. So that's why this is so important to monitoring wastewater. Here, this map tells you what's happening in the world. since the first report of the SARS-CoV-2 in wastewater in March 2020 from wastewater treatment plant around the Schiphol Airport in the Netherlands. Currently, there are more than 24 countries who is carrying out regional national monitoring program and to support the data from the CDC or public health reagents from the government. So firstly, this testing was carried out in the wastewater treatment plant, but now it has further expanded to different scenarios, such as care homes for near source tracking, hospital, school, hotel, or universities. So one of the successful example in 2020, Arizona State University has managed to test this SARS-CoV-2 in wastewater and then give an early warning for the students to move out and minimize this infection. However, in the analytical process, how can we test those wastewater samples from how can we test the SARS-CoV-2 in wastewater samples? So this is a typical workflow. As you can see that you need to collect samples from wastewater treatment plant and do the process control and do the sample processing. Then you run extraction of this nucleic acid from wastewater, run the PCR as you're testing in clinical samples. So this is a quite long process, which may take over 24 hours with the turnaround. The question is, can we really testing wastewater on the side of the sample collection in order to provide this immediate and valuable information to the local government agent in order to build up these early warning systems? So here, paper microfluidic device might be a solution. So that's would be more sensitive, fast, and able to carry out the testing in the field. However, there is a big difference between the wastewater samples and the clinical samples. One big difference is the sample size of wastewater usually needs milliliter scales. Typically, it's 50 milliliter. However, clinical sample only needs microliter levels. So, and also those SARS-CoV-2 particles has extremely low concentration in wastewater. That's needs as sample processing steps. So several years ago, we have developed this field-based kits for the extraction of nucleic acid from wastewater. So basically, we're using a syringe filtration systems to enrich those pathogens and using magnetic beads to extract and purify this nucleic acid material, and then carry out LAMP isotome amplification to test this genetic material. Then we can using a lateral flow device to read out these results. So this is the data we carried out as a proof of concept study in a local wastewater treatment plant. Based on this sample enrichment method for wastewater and using our paper-based platform together with Cas-RT-LAMP systems, we managed to test those SARS-CoV-2 in wastewater samples. So we're using either calorimetric dye as a readout or using lateral flow to test those samples to show the feasibility of point of need analysis. So the whole mechanism is that in the presence of SARS-CoV-2, quantities of amplicants are generated via RT-LAMP, which could actively the transcleavage of the Cas-12a guided RNA complex. Then the complex unspecifically and rarely cleaves the single-strand probes. The fluorescence is generated due to the separation of the fluorescence and the quench in the fluorescence method. So, however, in the lateral flow method, we can also have the strong test strand band is produced due to the separation of the dye and the biotin, because the former could combine within the modified GMPs and becomes immobilized on the test lines, while the latter is fixed on the control line. So based on this one, we can either using the visual dye for the detection or using the lateral flow device for the rapid testing device, especially useful for the diagnosis purpose. Here is the results, shows our sensitivity using this CRISPR-Cas-enabled molecular assay together with paper microfluidic device for testing of different concentration of SARS-CoV-2 in wastewater. So as you can see that we can either using the fluorescent dye or using electrofluid device to test these signals. And then we can normalize to allow semi-quantitative detection results. Based on this proof of concept study, we also carry out the field testing in the quarantine hotel over the summer in the UK in the UK. So we basically test a week and carry out the sample from 5 sites of a quarantine hotel from Monday to Friday. So we test 3 targets of the SARS-CoV-2 gene sequence, and which shows the different sensitivity depending on the samples. And we found that within the combination of this sequence, it can improve the sensitivity for this wastewater surveillance. And also, typically N gene has more sensitivity than S genes. That's because it's more stable in the wastewater. And finally, to give a quick sum up, we have already shown this Paper-Origami device as a sensing platform for the diagnostics and wastewater testing for public health. So we basically develop a few protocols for different sample detection, including clinical samples such as semen, blood, water, wastewater, and we also show the feasibility for different pathogen detection from SARS-CoV-2, influenza, E. coli, or salmonella for the water contamination or veterinary diagnosis, as well as a human infection disease such as malaria or the TB. So the whole testing platform is less than 45 minutes, and it's very cheap, less than £1. But it provides specific detection within a point-of-care application. It can also highly multiplexed for different pathogen detections. However, to address the pandemic challenges, we need to combine different technologies such as big data or machine learning techniques to real-time monitoring this prevalence of infection disease within the population, from the neighborhood levels to the population levels. Finally, I just want to show you this is kind of our contribution on this wastewater surveillance work. We quickly partner within UK academic to contribute to the National Wastewater Surveillance Program, and also to attract a lot of attentions. We published one of the first paper on this paper-based device for SARS-CoV-2 detection in wastewater, and dating back to March 2020. And it's also featured in the Science magazine after the first testing positive samples from the wastewater in Paris. So now this paper device has been displayed in London Science Museum. If you are interested, you are very welcome to have a look. Finally, I just end up with my talk within a small video from the BBC interview.
Now, a way of spotting COVID outbreaks much more quickly is being developed by scientists at Cranfield University in Bedfordshire, and the answer lies in, well, something none of us particularly want to think about: human waste. Our science correspondent Richard Westcott reports.
The key to controlling COVID outbreaks is to spot the virus as soon as possible, and this is where you'll find clues that it's spreading: at the local sewage works.
Now, it's not the most pleasant thought, but one way that the body gets rid of the coronavirus is through feces. And that means that by testing sewage, you can track down outbreaks in towns and villages. And that is where this clever, simple little device comes in.
Scientists at Cranfield University are developing a paper test to find COVID in our waste. Using chemicals and filters, it can spot genetic material left behind by the virus. But it's inactive, so they say you won't catch COVID from the water. It's designed to be cheap, easy for anyone to use, and gives a result in an hour. Green lights means there's COVID around.
It can be widely used as a routine test. For example, the schools and care homes, or even hospitals, or even hotels, leisure center, like some public places, to monitoring this wastewater.
Up the road, they're testing at the university's own sewage works. They say the technique could find hotspots much sooner than current swab tests.
Okay, finally, I would like to thank very much for my founders, collaborators, and my students who contribute a lot of this work. Thanks so much for your attention.